Alrighty, so what we're going to do today is acetyl-CoA transport as well as some mitochondrial permeability stuffs. So really quickly, we know that the mitochondrial membrane has a double layer of plasma membrane. Certain things can cross it, some things cannot. The inner mitochondrial membrane is impermeable. The outer mitochondrial membrane has some perforations in it, allowing more stuff to cross, but we're not really worried about that. What we are worried about is what we can actually get across the plasma membrane and what cannot. So, going back and kind of encompassing as many things as possible, there are transporters on the mitochondrial membrane, or there are pathways by which these things can get across. The mitochondrial membrane is permeable to the following things, which I need to find now. They are permeable to malate. We learned about that. That's going to be covered extensively in acetyl-CoA transport. It's also permeable to pyruvate, as well as phosphoenol pyruvate, PEP, phosphoenol pyruvate. These two are components of gluconeogenesis. That's where we kind of know them from. We also know that the mitochondrial membrane is permeable to citrulline and ornithine. These are intermediates of the urea cycle, which cross over the mitochondrial membrane. Alright, that's pretty much everything that we really need to know that the mitochondrial membrane is permeable to. It is impermeable to a couple things such as NADH. Any really high energy thing, NADH, FADH2, you got your NADPH, nothing's really going to cross that plasma membrane. This is the same with acetyl-CoA. We have this really high energy portion right there that's not going to be able to move across the plasma membrane. The mitochondrial membrane is also impermeable to oxaloacetate, which is good to remember. We're missing something in the permeable membrane. What might that something be? That something would be citrate, which is ironically what we're going to be using to get acetyl-CoA across the membrane, which was the purpose of this video. Haha! Alright, so we've got this pretty well, so we're going to move on to... It's just a little bit of an intro. Now we're going to actually do some acetyl-CoA transport. So, really quickly, we know that acetyl-CoA is produced in the mitochondria by a couple different pathways. Those pathways can essentially be summarized into the pyruvate dehydrogenase complex, the PDH or pyruvate dehydrogenase complex, as well as the, what's it called? Amino acid catabolism. Alright? We have several amino acids that will break down into acetyl-CoA or acetoacetyl-CoA. Those amino acids are leucine, here, leucine, lysine, where else are they? Leucine, lysine, phenylalanine, which is going to break down into tyrosine, which can be broken down into acetyl-CoA, as well as threonine. So just kind of a tying everything together kind of idea. These are going to form acetyl-CoA via mechanisms that we're not really going to discuss right now because that would take way too long. Now, we just went over the fact that acetyl-CoA cannot cross the plasma membrane easily. So what we're going to do is we're going to combine it with oxaloacetate into something that can cross the plasma membrane. Alright? Now, if we are making a bond, we need to have some kind of energy. Creating a bond requires energy. Breaking a bond releases energy. Because we're making this bond here, we're basically taking acetyl-CoA, tacking it on, and creating citrate. The enzyme that does this is citrate synthase. Citrate synthase. One of those really nice enzymes. First name is the product, second name is what the enzyme is doing. It's making citrate. Now the energy for the creation of the acetyl bond to the oxaloacetate is going to be from this high energy bond right there. Hydrolysis of that coenzyme A bond is going to provide the energy to create citrate from oxaloacetate and that acetate group. So we're going to have a release of coenzyme A-SH because we've hydrolyzed that sulfide bond between the coenzyme A and the acetyl group. We now know that citrate can cross the plasma membrane. It was one of those wonderful permeable things, which I forgot about until the very end. So we now have citrate in the cytoplasm. Now citrate in the cytoplasm, once it's there, is going to be broken down pretty much instantaneously, and it's going to form two things. It's going to form acetyl-CoA and oxaloacetate. Now, oxaloacetate. It's going to form those via the action of the enzyme citrate lyase. Now citrate lyase, we are breaking a bond, however, we also need to make acetyl-CoA, right? Our entire purpose of doing acetyl-CoA transport is to obviously transport acetyl-CoA across the membrane, right? Across the mitochondrial membrane. Therefore, we need a coenzyme A, right? And because the bond here is a very high energy bond, we're going to need the input of energy to create that bond. I know we are breaking a bond. We are breaking the bond between the acetyl group and oxaloacetate, but that bond does not have enough energy to create the sulfide bond to the coenzyme A. Therefore, we're going to need the input of ATP. ATP goes in, we're going to get out some ADP and some inorganic phosphate. Now, acetyl-CoA right here can be used in whatever pathway you really need. We are doing acetyl-CoA transport because we have an excess of energy. We have too much energy for the body to use. Otherwise, acetyl-CoA would keep running through the citric acid cycle until it was all used up. Now, because we have too much energy, we're getting that acetyl-CoA into the cytoplasm to create some kind of storage form of energy. In this case, that storage form would be as a fatty acid. We'll go over biosynthesis in another video, but for fatty acid synthesis, you require NADPH. All right? Keeping that in mind as we move forward here, what's going to happen to oxaloacetate is it's going to be dehydrogenated because we know that oxaloacetate cannot cross that plasma membrane. However, it can take one step to create a compound that can. That compound is malate. The enzyme that does this is malate dehydrogenase. And we know that the opposite reaction occurs in the TCA cycle. So we can basically say that this happens here, bam, malate. We know the mitochondrial membrane is permeable to malate, so malate crosses and it can form oxaloacetate via the exact same enzyme, malate dehydrogenase. Now this is an example of an isoenzyme, an enzyme that is present in two different cellular compartments. This right here is going to be your mitochondrial malate dehydrogenase. This right here is going to be your cytosolic malate dehydrogenase. Not super important. Now we know that in the citric acid cycle, this is the direction of the reaction. The citric acid cycle is used to produce energy. You're producing energy from the electron transport chain via the input of electrons from electron carriers. So we know that this reaction here is going to create an electron carrier. Therefore, it's going to create NADH. That is what the citric acid cycle is designed to do. That means that over here in the reverse reaction, we are actually going to use NADH. It's kind of alright because everything really balances out in the end. You use one here, create one there, life is kind of happy. Alright, now this is assuming oxaloacetate to malate, malate moving across the membrane assumes that we have an excess of energy as well as enough NADPH to make fatty acids from the acetyl-CoA that we just transported. In the case that we do not have enough NADPH, we're going to do something else with this malate. All right? Just as a quick aside, an excess of energy from carbohydrates will create NADPH via the, what cycle is that now? Pentose phosphate pathway. There we go. Via the pentose phosphate pathway. Now, the pentose phosphate pathway relies on glucose 6-phosphate as the initial metabolite. If you do not have glucose 6-phosphate, you cannot run the pentose phosphate pathway. In the case that we have an excess of energy from something other than carbohydrates, when we break that thing down, we will not make glucose 6-phosphate because we have an excess of energy. We're going to be storing everything as fat. So as soon as all of the carbohydrate is used, we will not have any more glucose 6-phosphate to run the pentose phosphate pathway. Instead, we need to get our NADPH from somewhere else. That somewhere else is going to be the malic enzyme. This enzyme is activated in the cytoplasm and breaks down malate into pyruvate. All right? We know that malate is a C4 compound and pyruvate is a C3 compound, so it's going to release some carbon dioxide. We also know that because of this enzyme, we are going to be making NADPH. This NADPH is going to be used for fatty acid synthesis and requires the NADP+ in order to tack that hydrogen on. So that's kind of cool. We also know that the mitochondrial membrane is permeable to pyruvate. So this pyruvate can flow right across that membrane, right till here. And then via the first step of gluconeogenesis, it can be acted on by pyruvate dehydrogenase, pyruvate carboxylase, mind you. Let me check and make sure I'm right on that. Yes, by pyruvate carboxylase, which is going to use some carbon. I've seen it in textbooks as both of these things. So if she asks for a source of carbon, you can fairly easily do both of those. If you get it wrong and have HCO3, give me a shout. I'll argue it for you. Because I'm probably going to use HCO3. It seems more accurate. So we're going to use that, and it's going to require an input of ATP, as we know from our, as we know from our gluconeogenesis learnings. All right. So this looks a little bit complicated. As a last point, we now have our acetyl-CoA that can be used for fatty acid transport. We have NADPH if we have an excess of energy from something other than carbohydrates, probably amino acids. Amino acids, hey! So, all of the things in black, that's the pathway that's going to happen if you have an excessive amount of energy from carbohydrates. Carbohydrates giving you an excessive amount of energy. All of the red is if you have an energy excess from something like amino acids. Those amino acids will not give you glucose 6-phosphate to run the pentose phosphate pathway. Therefore, the only NADPH that we know that can be produced is via the malic enzyme. Thanks so much for watching. Have yourselves a great day.